Hello everyone, today I'll be presenting my iSTEM Scholar Final Symposium. My name is Baoanh Nguyen and I'm from Houston, Texas. I'm currently a junior at Langham Creek High School and my STEM interests are mainly in integrated biology relating to environmental sciences, evolutionary biology, water quality, and anatomy and physiology. This summer, I had the opportunity to be a part of the Bernal Lab from Purdue University and learn more about the field of ecology and evolutionary biology. My PI was Dr. Ximena Bernal, and my research mentors included Desi, Kathy, and Ethan. In the Bernal Lab, they focus on the evolution of animal communication, especially in regards to frogs and flies. To be more specific, they investigate the exploitation of neuron communication systems. This is when unintended receivers, such as predators or parasites, eavesdrop on mating signals produced by, for example, frogs or toads. And these predators and parasites exploit this communication system in order to detect their victims. These unintended receivers are also called eavesdroppers. In a specific example, a specific eavesdropping interaction that they examine is between the frog-biting midges and the tungura frogs, in which case the female frog-biting midges eavesdrop on the mating call of tungura frogs to locate and draw the frog's blood. The Bernal Lab also looks at the consequences of this interaction, such as the transmission of trypanosomes, which are parasites, from the midges to the tungura frogs. And another eavesdropping interaction that they study is between tungura frogs and frog-eating bats, who, like the frog-biting midges, eavesdrop on the mating calls of frogs, except not to draw blood, but rather to feed on the frogs. In order to understand how eavesdroppers develop traits associated with this behavior, they also look at the evolutionary ecology of eavesdroppers. Additionally, the lab studies how frogs and other organisms effectively communicate in an acoustic space. And they also examine how behavior and communication possibly enable invasive neurons to thrive in foreign environments. And they look at the effects of urbanization on animal communication networks, especially with regards to neuron interactions with both unintended and target receivers. During my time in the Bernal Lab, I gained a multifaceted experience in a variety of areas pertaining to being a part of a lab. In addition to understanding how to read scientific papers, I was able to explore methods to effective science communication, learn a little about statistics, and participate in research. First, I'll give a brief summary analysis of the scientific papers I read this summer. So first, we read and discussed a paper that revisited the drivers of acoustic similarities in tropical neuron assemblages. We agreed that the paper could have been written more clearly, but overall, the paper re-examined the factors that influence the acoustic space of mixed species assemblages from an evolutionary lens. And these factors include phylogenetic relatedness, body size, habitat, and environmental filtering. Next, we discussed the paper over systemic inequalities for LGBTQ professionals in STEM, which came to the conclusion that LGBTQ STEM professionals were more likely to experience disadvantages, such as limitations in opportunities, harassment in the workplace, and professional devaluation than their non-LGBTQ coworkers. The paper also found a trend that LGBTQ professionals tended to suffer frequent health difficulties and thus were more likely to leave the STEM field. We also discussed the paper on sampling biases and how they shape individuals' view of the natural world. This paper attempts to address pervasive sampling and observation biases in studies on biodiversity and how these biases have affected our knowledge of life on Earth and thus influence our ability to protect species. After that, we analyzed the paper on science communication that emphasizes inclusion, equity, and intersectionality. It underscores the importance of a multi-scaled inclusive science communication that recognizes varying identities, acknowledges the experiences of historically oppressed communities, and fosters public engagement and a sense of belonging in STEM. The paper also provides a model for advancing inclusive science communication. And independently, I have read a paper on the cues that frog-biting midges use to find the neuron hosts I have used this paper for a couple of science communication projects in my lab and for my blueprint literature review. I've also independently read a paper on changes in the mating calls of frogs in urban settings, possibly due to adaptive responses. And throughout the course of the program, I was introduced to the strategies to effective science communication. This included taking the audience into account when constructing a presentation, identifying the goal of the presentation, and avoiding jargon. I also learned methods to incorporate inclusivity in science communication. And to apply this knowledge, I worked on 2 science communication related projects. For the first project, I was instructed to create a TikTok video that summarized findings from a scientific paper. And although I had a few difficulties in translating the data without using jargon, I was able to gain firsthand experience in science communication. Currently, I'm working on my second science communication project, in which I've chosen to create a video presenting findings from 2 scientific papers to a general audience. And in addition to science communication, a couple of my research mentors introduced me to statistics in a biological context. From this, I was able to have a basic understanding of the null and alternative hypotheses, the p-value, the mean and standard deviation, the normal distribution, as well as the difference between categorical and quantitative graphs. And I also learned to distinguish not statistically significant data or graphs. Finally, throughout the course of my time in the Bernal Lab, I had the opportunity to participate in their research. As a human proxy to how predators perceive toads, I analyzed images of varying species of toads in their natural environment and rated them on a scale of 1 through 5 based on whether they were cryptic or conspicuous in their surroundings. A rating of 1 would be cryptic and a rating of 5 would be conspicuous. My ratings, along with the ratings of other human observers, would be combined in a composite measure, and the mean value across observers would be used for each species. However, a second measure involving computer-generated estimates would also be used for comparison, and in the end, these measures are used to identify evolutionary shifts in coloration for toads. And lastly, I would just like to give a big thank you and shout out to all of the members of the Bernal Lab, the entire iSTEM Scholar team, and the Blueprint team for being so understanding and flexible. And thank you for teaching me so many things about college, lab work, and research and presentations. And thank you to everybody who has supported and encouraged me throughout this program. I really enjoyed my time in this program because of all of you. Thank you so much.